Hello, students. Thank you. Welcome back. We're going to start a new topic today. We've completed the last topic. We're going to start today with proteins. Proteins, part 1. Proteins, part 1. Okay, let us begin. Just a little review about proteins here. Some background information about proteins themselves. I need a solid color here for the background. I kind of wrote on my background, can't do that. Okay, if you have a cell, let's say we have a cell here. Let's say here we have a cell. This cell is going to have lots of things in it. It's going to have water in it. It's going to have proteins, nucleic acids, carbohydrates, and lipids. All of these components are going to be in this cell. And if I ask you the question, what percentage of the dry weight of what substance of these 5 substances that are in the cell, which one do you think is present in the greatest amount? The water, proteins, nucleic acids, carbohydrates, or lipids? Which one is going to be present in the greatest amount? Water, exactly. So water is going to be present in the greatest amount. Now, what if we could somehow take all of the water out of that cell? Now there are going to be our 4 macromolecules are going to remain. The proteins, nucleic acids, carbohydrates, and lipids. Of those 4, which will be present in the greatest quantity? Has to be proteins, since that was the title of the last slide. So it has to be proteins. Exactly. Now, how will we go about removing the water from this cell? Yeah, we could place it into a hypertonic solution, but then how will we get the hypertonic solution then away from? How we get the hypertonic solution away from the cells? That's a good answer. What was that? Yes, that would be the easiest thing to do. Now, even if we did put it in a hypertonic solution, you would lose a lot of the water, but it would still be wet. And so, a better way to get rid of the water is to put it in this thing called a drying oven. Okay? We put it in a drying oven. We dry the cells out, and then once you dry the cells, then you would be left with, the cell would have just proteins, nucleic acids, carbohydrates, and lipids. And you would not have any water there. What percentage do you think, now we said the proteins would be present in the greatest amount. What percentage of this cell do you think would be protein? Well, of the dry cell, what percentage of the dry cell would be protein, of the cell's dry weight? Well, it has to be greater. Well, I did say proteins are present in the greatest amount. So, if proteins were 33%, then the other 3 would be like maybe 20%. But actually, the protein concentration or the protein percentage is actually more than 50%. The amount of protein is greater than the other 3 combined. Okay. This makes sense? That's the first concept I need you to know about proteins. Proteins make up a very large amount of the cell. Why do you need proteins in cells? What do proteins do? And it's not even on my notes. Exactly. There's a specific function for everything that we do. There's an enzyme there are enzymes that add OHs to tyrosine to make dopamine or to make dopa. There's enzymes to do everything. There are pigments that make your hair color, your eye color. There are proteins for to make actin and myosin to give you contraction. So proteins do all of the things that make you you. Okay. Let's continue. Now that we know how much protein is in cell. Now let's talk about how we make a protein. Where do proteins come from? Why are your proteins different than your proteins? Or why are your proteins different from your proteins? What makes your proteins different? The DNA. Exactly. Within a cell, you have this polymer. Okay, and this polymer is shaped like this here. And within this polymer, you're going to have, on this polymer, you're going to have some nitrogenous bases. I'm going to kind of a lighter color. I need to really be able to see that. What's a good color? Yes. You're going to have nucleotides on DNA, on this DNA backbone. And these nucleotides, the sequence of these nucleotides determine what amino acids go into your proteins. And that's what makes one person's proteins different from another person's protein. Okay. Then you're going to have nucleotides here. And so this molecule, what is this molecule called? What's the name of this molecule? DNA is the name of this molecule, exactly. And it has the shape of a double helix. This is DNA. Okay? Now, DNA is in the nucleus of the cell. Okay, and so the DNA comes out of the nucleus of the cell. Then the ribosomes read the DNA to make the protein. Does that make sense? Some of you all aren't listening to me because that was so false what I just said. DNA, I said that DNA comes out of the nucleus. The ribosomes read the DNA to make your proteins. Is that true? No, that's not true. Not true. So DNA makes a functional copy of itself. What's the name of that molecule that's the copy of DNA? It's RNA. It's going to be RNA. And so here we're going to have this single-stranded molecule is going to be called RNA. And it doesn't make an entire copy of the DNA. It just makes a certain segment of the DNA. The segment, good question for you all. What's the name of the segment of DNA that goes to make the functional RNA? What's the name of that DNA segment? Okay. So now if you have a pigment, like if your eyes, let's say your eyes will have a hazel color. There is a pigment, in your DNA, there's a section of DNA that codes for that hazel pigment. What is the name of that section of DNA that makes that hazel pigment RNA, that makes that hazel pigment protein? No, that's something else we'll talk about, the Okazaki fragment. That's something else we'll talk about later. That has to do with the leading strand and the lagging strand. That will be later. The replication fork, that has to do with DNA. You're all talking about DNA replication right now. And I just need to know what's the name of what's the name of the segment of DNA that gives you your traits, your eye color? A gene. Exactly. It's just a gene. Exactly. And so here we're going to have a gene here. And so that gene of DNA is going to make a functional copy of itself, RNA. Now, can the RNA leave the nucleus to go to the cytosol? No. Messenger RNA can leave the nucleus. tRNA is involved in translation. You all are getting way ahead of me here, I tell you. But it is involved later on. Remember, the tRNA just holds the amino acids. Okay? And so here, the messenger RNA is going to leave the nucleus, and then once it gets to the nucleus, there are going to be some proteins in the nucleus. Okay, and then these proteins, they're going to read the DNA they're going to read the RNA, and as they read the RNA, then they're going to put together the right sequence of amino acids to make our.
Can you say that one more time?
Okay.
So you make a copy of the RNA.
Okay.
The messenger RNA, and then what?
Okay. Her question was, can you repeat this again after you make a copy of the RNA? So I'm going to just start from the beginning. Okay. You have on your DNA. DNA is a very long strand. It's about if you stretch DNA out in a cell, it's about 1 meter long. But we're going to take a very small portion of that called a gene. That gene, DNA is going to make a copy of that gene into messenger RNA. And that's still in the nucleus. Now the messenger RNA is going to leave the nucleus, go to the cytosol, and there are going to be these proteins in the cytosol that are going to read the messenger RNA and then put the right amino acids together to make our polypeptide. And so this is going to be our polypeptide. Everyone with me so far? Now, I have two questions for you before I move on to the next slide. The next image. What is the name of the protein here? This protein here. Let me make sure I can grab it. What's the name of this protein here that reads the messenger RNA to put the amino acids in the right order? Ribosomes. Topoisomerases have to do with how tight, how cold the DNA is and uncoiling the DNA. But we'll talk about that later on. And so here we're going to put, this is going to be our ribosome. Okay. Now the ribosomes will read the messenger RNA and put the right sequence of amino acids together to make our polypeptide. And now we have a polypeptide. But when I first started this slide off, we weren't trying to make a polypeptide. What were we trying to make? A protein. So what's the difference then between just the polypeptide and the protein? No, not ATP. Okay. Say that again? Okay, proteins have 4 levels of structure sometimes. That has something to do with the answer. Okay then. I'm going to make this longer. Okay, now it's still a polypeptide. Okay. It has to have alpha helices and beta sheets. The answer is exactly. A polypeptide is just a long strand, but a functional protein, is folded in the correct way. So a polypeptide is an unfolded protein. And so all you have to do now is just get some folding proteins to come in and help this protein to fold correctly, and then you have a protein. Everyone okay with that? Now, one more question for you. What is the name of the process by which you make messenger RNA from a gene? What's the name of that process? Transcription. Exactly. Oh, very good, very good. So let's put here So when you start out with a gene from DNA and you make a functional copy of that gene into messenger RNA, then that process is called transcription. Everyone okay with that? That's making messenger RNA. What if you have messenger RNA, but you want to turn that messenger RNA into a polypeptide, putting amino acids together to make a polypeptide? What's the name of that process when you go from messenger RNA to a polypeptide? Translation. Very good, you all. Very good. Okay. Does this slide should make sense now? Does this slide make sense? You look confused. What's the name of the process? Oh, her question was, she missed the last thing that I said. The question was, what is the name of the process by which you take messenger RNA and make a polypeptide? What's the name of that process? And that answer was translation. Translation. Now, why, just from a functional standpoint, why do you need to take your gene and make messenger RNA? Why can't the ribosomes just come in, or why can't the ribosomes just read the DNA? Why go through this extra step? This is an intuition question. Why? Why go through that extra step? Does everybody understand the question? Okay. Yes. What the ribosomes do, the ribosomes read the messenger RNA to make the proteins or the polypeptide. Why don't they just skip that step? Why not just skip the middleman? Why don't the ribosomes just go to the DNA, read the DNA to make your... Oh. Okay, it wouldn't know the right sequence. Say that again. To conserve the DNA, so that might... Oh, that might destroy the DNA. Okay, that's reasonable. Okay, that might... You don't want to open the DNA up, because every time you do that, you have to open the DNA, and that could damage the DNA. That's a reasonable answer, but you have to open the DNA up anyway when you make the messenger RNA. Right, yeah, yes, but why not just leave the C? Why not just leave the T there and just forget the U and all of that stuff? See, what I wanted to get to you all is this: in a cell, this is what I want. Maybe that wasn't the best way to approach this, but I want you to get this understanding here. Here we have the nucleus. This is the nucleus. Let's say this is a nucleus of a cell, and in this nucleus we have our DNA. Okay? And within this nucleus you have some holes here. And out here you have your ribosomes. Now, two things here. The ribosomes could possibly, I didn't draw them to scale, the ribosomes could possibly come into the nucleus, read the DNA, and then make your proteins. But then all of your proteins are going to be, that you make, all your new proteins are going to be stuck in the nucleus. And so you don't want that to happen. And also, these pores here, when you make if this is your messenger RNA here, this messenger RNA can just come through these pores and go to the ribosome. But DNA is just way too big. The DNA would never be able to get out of these pores. And also, when you get outside of the nucleus, if you get outside the nucleus, there are lots of nucleases out here. And they like to destroy DNA. And so they like to destroy any type of nucleic acids. And so if the DNA, first of all, the DNA can't go through these pores, and if it did, it would be destroyed. And you don't want that to happen. So you want to keep the DNA as protected as you can, because you don't want it to ever come outside of the nucleus. Does that kind of make sense? And so there could be a million answers for this, but I just want you to understand somewhat of the teleological rationale. Okay? Everybody okay with this? Where proteins come from. Okay, excellent. You all are. Let's talk about some different types of proteins. If you would, please, I want you to just zoom out on that camera for a second here. Zoom out. Let me have 1, 2, 3. Let me have the three of you all. 1, 2, 3, 1, 2, 3. 1, 2, 3. Come and stand up here. McCafferty, let me have you, and let me have you. You all just stand on the side for a second. There are two. Okay, let's make sure he has enough. So just zooming out so you can see those. Okay, you stay there. You stay right there for just a second. Okay, you have them? Okay, there are two types of proteins. There are structural proteins, and there are going to be functional proteins. And structural proteins are kind of long like a rope. So I want you all to make yourself into like a hold hands. Just hold one hand. Yes. Now stretch yourselves out. Okay, very good. Now they're like, can you see them? Oh, that's not like this. Okay, can you see them? Okay, now they're like a rope. I want you to grab her arm. Okay, I want you to grab, you grab her arm, both hands. Yes, her hand, or yeah. Okay, now you all try to pull them and stretch them. Try to pull them and stretch them. See, can you see that? Okay, that's okay. But the point is here, he can't really pull them anymore because they're structural. They're long like a rope. Does that make sense? Okay, very good. Now, I want you all to do this. Oh, three of you all still hold hands, those hands. Now, you all make yourselves into a ball. Make yourselves into a ball, but don't join that other hand. Okay. Okay, now they're a ball like a globular protein. Now they're like an enzyme, because enzymes are round. Does that make sense? Okay, now enzymes, they can do functions, they can do things, they can function, but they can't, they aren't good for structure. Now I want you to grab her other arm. Now I want you all to try to prevent them from pulling you apart. Okay, you grab her arm. Okay, now pull them apart again. Try to stop them. But they shouldn't be able to if you guys have any level of strength, guys. And so, the point is here, if it's a globular protein, you can stretch it very easily because it's not designed for structure. It's just designed for function. Does that make sense? Okay, very good. Thank you all. Thank you all. Excellent. Oh, I forgot to change it so you can see that. Because you all won't be able to see that now. Well, unless you watch it on the video. If you watch it on a computer, you'll be able to see it. See, that's why I don't really like this. I should have used something else. But that's okay. Okay, let's look at the two types of proteins. Let's look at the two types of proteins here. There are structural proteins which look like they're long like fibers. And so this is a structural protein. Structural proteins are long like fibers. And if you look at the DNA, the protein strands themselves, the different proteins is going to look something like this. And so these proteins have a lot of tensile strength. What is the difference between tensile strength and compression strength? Does a rope have a lot of tensile strength or a lot of compression strength? It has a lot of tensile strength. What does that mean? Right. What is tension? What is a tension force? Is it a pushing or a pulling force? It's a pulling force. So structural proteins have a lot of tensile strength. This bench here, does it have a lot of tensile strength or a lot of compression strength? It has a lot of compression. You could probably put a lot of weight on this and it would be fine, but it doesn't have a lot of tensile strength. Okay? And so these proteins that have this type of, that are like ropes, these are called fibrous proteins. Okay? Now we've talked about in signal transduction, we've talked about some fibrous proteins. Can anyone name a fibrous protein? I'm going to draw something. Exactly. Myosin or actin. Those are tensile proteins because they have structural properties. They pull to give you contraction. Make sense? Excellent. Okay, did you zoom all the way out on that? Did you zoom back in? Okay, I can zoom out on it. Okay, put your notes away.